Dear students, I welcome you to the e-learning program through ePG Pathshala. I am Dr. K. Pandima Devi, working as Associate Professor in the Department of Biotechnology, Alagappa University, Karaikudi. In this session, I am going to explain to you about the classification and types of the different antioxidants. I will be explaining about the significance, classification and types of antioxidants, which are the molecules that help a cell to protect against the deleterious effect of oxidative stress. As you know, oxidative stress has been considered as the important factor in the pathophysiology of different diseases, including cancer, neurodegenerative disorders, diabetes and so on. We will discuss about the different types of antioxidants which aid the system in neutralizing the effect of free radicals. Antioxidants, classification and types. After completing this module, you will be able to understand about the significance of antioxidants in protecting the body, know the different types of antioxidants available, and also appreciate the method of classification of antioxidants. Before seeing in detail about the antioxidants, we will first know about what free radicals are. As you are aware, cells are made up of molecules which consist of one or more atoms of one or more elements joined together by chemical bonds. When a weak bond splits, it leaves an unpaired electron in the atomic orbital, which is called as free radical. Example of oxygen containing free radicals include superoxide anion radicals, nitric oxide radical, singlet oxygen and hydrogen peroxide. Though these oxidants are formed as a normal product of aerobic metabolism in a cell, when produced at a higher level under different pathophysiological conditions, these highly unstable and reactive free radicals are capable of damaging the macromolecules of the cell like lipids, proteins, carbohydrates and DNA. However, to protect against the harmful effects of these free radicals, the cells have a protective molecule which is called as an antioxidant. So these protective antioxidants are broadly defined as molecules with ability to prevent damage to cellular components arising as a consequence of chemical reactions involving free radicals. The antioxidants act in different ways, like terminating the chain of free radical reactions by getting oxidized by itself or removing the free radical intermediates, chelating the elements like metals which are involved in the production of free radicals, and enhancing the expression of genes coding for antioxidant proteins. The antioxidants can be classified into different types based on their properties, source or functions. Some of the broad methods of classifying antioxidants include water and lipid soluble antioxidants, which is based on solubility, natural and synthetic antioxidants, which is based on the source, enzymatic and non-enzymatic antioxidants, which is based on the properties, endogenous and exogenous antioxidants, which is based on the source, and primary or chain breaking antioxidant and secondary or preventive antioxidant, which is based on the function. However, these classes of antioxidants have an interlinked network because some of the endogenous antioxidants may be either enzymatic or non-enzymatic, and some of the exogenous antioxidants may be either natural or synthetic. But to give an overview on the different methods of classification of antioxidants, we will be discussing each of the classes separately. First, we will see about the types of water and lipid soluble antioxidants. Water soluble antioxidants, as the name implies, are hydrophilic in nature, and the common examples include ascorbic acid, glutathione and uric acid. In a cell, they will be present mostly in the cytosol, whereas glutathione is present in extracellular environment also. Now we will see some of the examples of water soluble antioxidants. One example is ascorbic acid. It cannot be synthesized by humans, hence should be supplied through external dietary source. It is an exogenous antioxidant for humans. It is found in fruits and vegetables and is a very effective antioxidant of human plasma. It acts as an electron donor. On donating one electron, it becomes a free radical named as ascorbyl free radical. The donated electron is transferred to the harmful free radical and converts it into non-radical form. The ascorbyl free radical, which is formed in the due course when compared to the other toxic free radicals, is fairly unreactive. It is for this chemical property that ascorbate is preferred as a good free radical scavenger. Vitamin C increases the antioxidant power of vitamin E. Though vitamin C is a hydrophilic compound, it protects the hydrophobic membrane lipids through the regeneration of vitamin E. The water-soluble antioxidant glutathione is a tripeptide made up of cysteine, glycine and glutamine. This cysteine-containing tripeptide is ubiquitously present in all cell types in humans. Its reduced form is called as glutathione GSH, and in oxidized form it is called as glutathione disulfide GSSG. It acts as an antioxidant by donating hydrogen atom and reducing the free radicals directly, or acting as a substrate for enzymes like glutathione peroxidase, which removes the free radicals like hydrogen peroxide. The water-soluble antioxidant uric acid is the end product of purine metabolism in humans. The last two reactions in generation of uric acid are catalyzed by xanthine oxidase. Since there is generation of free radical in the last two steps involved in the formation of uric acid, the levels of uric acid is generally considered as a biomarker of oxidative stress in conditions like hypertension. It is a singlet oxygen quencher and scavenges peroxyl radicals and hydroxyl radicals. By acting as a free radical scavenger, it protects the integrity of erythrocyte membrane. It is also a major antioxidant present in plasma, which contributes to up to 24% of free radical scavenging ability of plasma and protects the cells from oxidative damage. Apart from plasma, it is also present in extracellular spaces like synovial space, amniotic fluid, aqueous humor and vitreous body. Bilirubin, which is another water-soluble antioxidant, is a chain-breaking antioxidant. When bilirubin acts as an antioxidant, it gets oxidized to biliverdin, but it can be recycled back to bilirubin by biliverdin reductase. Now we will see about the lipid-soluble antioxidants, which are hydrophobic in nature and are normally localized on the membranes of the cells. Hence, they help to protect the cell membrane from lipid peroxidation. The common examples of lipid-soluble antioxidants include vitamins like vitamin E, beta-carotene, galates, resveratrol and ubiquinol 10. In this class of antioxidants, vitamin E, otherwise termed as alpha-tocopherol, is a chain-breaking antioxidant. Generally, the polyunsaturated fats present in the biological membranes are more prone to attack by free radicals like peroxyl radicals. In such instances, alpha-tocopherol donates its hydrogen from the OH group to the peroxyl radical and converts it into hydroperoxide product. Vitamin E, which is consumed during the antioxidant process, is generated by vitamin C. Since it is very effective in preventing peroxidation of lipids present in membranes, vitamin E is an important factor for maintaining the integrity of lipids in the membrane. Beta carotene, which is also fat-soluble antioxidant, is otherwise called as carotenoids. They are the most common natural pigments which are mainly taken by animals through diet. Beta carotene acts as a precursor to vitamin A, which is necessary for normal growth and development and also for maintaining the immune system and vision. It helps in the regeneration of the antioxidant vitamin E. It is also a chain-breaking antioxidant like vitamin E, but less effective when compared to vitamin E. It also effectively scavenges singlet oxygen free radical. The fat-soluble antioxidant ubiquinol 10 is an endogenously synthesized antioxidant which is present in most of the human cells. Though it is produced in our body, they are also taken as nutritional supplement. It is found mostly in dark leafy green vegetables like broccoli, spinach and so on. It has 10 isoprenoid units which contributes to the lipophilicity of the molecule. Because of its lipophilic nature, it inserts into the membranes and protects the biological membranes from lipid peroxidation. It is also involved in mitochondrial ATP synthesis and helps in the regeneration of other antioxidants like vitamin C and E. The second class of antioxidants, which is classified according to the source, is natural and synthetic antioxidants. The natural antioxidants are substances present in plants and animals which offer protection against the oxidative damage. Examples are polyphenols, vitamins, minerals, antioxidant enzymes and glutathione. Now we will see some of the examples of natural antioxidants. One example are polyphenols, which are the major natural antioxidants taken through dietary source. They are the secondary metabolites produced by the plants and they are not produced by humans. They are utilized by the plants for their normal growth and also offer a defensive mechanism against infection and injury. Depending upon their chemical structure, polyphenols act as a free radical scavenger by donating hydrogen atom or electron or act as a singlet oxygen quencher. Hence, intake of foods and beverages which are rich in polyphenols increases the antioxidant capacity of plasma. Many polyphenols are utilized as drugs by humans for treating oxidative stress associated disorders. The main dietary source of polyphenols include tea, fruit juice, vegetables, red wine, and even chocolates and cereals. Apart from this, plants which are not commonly used as foods, like herbal plants and seaweeds, are also good sources of polyphenols. Different classes of polyphenols are present, including phenolic acids, tannins, flavonoids, stilbenes, and coumarins. They have at least one aromatic ring substituted by one or more hydroxyl groups. Of these different classes, the class which has a remarkable antioxidant activity are the flavonoids. They have a phenolic benzopyrene structure. Over thousands of flavonoids have been identified in the plants which have been proven to possess antioxidant activity. There are different subclasses of flavonoids including flavonols, flavones, flavonols, flavonones, isoflavones and proanthocyanidins. Flavonoids have functional hydroxyl groups in them. The free radical scavenging or metal chelating ability of flavonoids depends on the degree of hydroxylation, substitutions, and so on. Hydroxylation at the B ring has been linked with the antioxidant activity, especially flavonoids with 3' and 4' hydroxylation act as very good antioxidants. The flavonoids which act as more effective free radical scavengers can be obtained through different sources. For example, quercetin is obtained from buckwheat, onion and sweet potato, and the flavonoid catechin is present in more amount in tea leaves. Of the different minerals which are obtained naturally, selenium, which is an essential trace element, acts as a good natural antioxidant. It triggers the antioxidant enzymes like superoxide dismutase, catalase and glutathione peroxidase and increases the total antioxidant activity of the cell. This in turn helps in protecting the DNA from reactive oxygen species mediated damage. It also inhibits the metal mediated DNA damage. Selenium is also present in the active form in certain enzymes in our system called as selenoenzymes. In the selenoproteins, it is present as the amino acid selenocysteine. The most important selenoenzymes are the reactive oxygen species decomposing antioxidant enzymes like thioredoxin reductase, glutathione peroxidase and so on, which have a functional role in protecting the cells from oxidative stress. Apart from the selenoproteins, in the biological system, selenium occurs in other chemical forms also, including selenomethionine, inorganic selenides and selenates. Vitamins like vitamin A, E and C obtained from natural sources like plants are also examples of natural antioxidants. The natural antioxidant melatonin is a small molecule hormone derived from the amino acid tryptophan. It stimulates the antioxidant enzymes and also the synthesis of the antioxidant glutathione. It acts synergistically with the other antioxidants like vitamin E, vitamin C and glutathione. Other examples of natural antioxidants include antioxidant enzymes, glutathione, ubiquinol 10 and even uric acid, which is present endogenously in humans. We have seen about natural antioxidants. Now we will discuss in detail about synthetic antioxidants, which are not found in the nature and are usually synthesized and used as a substitute to the antioxidants obtained from the natural sources. They are most commonly used as preservatives in packed food, which are stored for longer time. To protect the animal fats in the packed food from rancidity, which is a type of spoilage, antioxidants are generally added, which slow down the rate of oxidation and increase the resistance of fats present in the foods to oxidation. The most prevalent synthetic antioxidants are butylated hydroxyanisole, tertiary butyl hydroxyquinone, butylated hydroxytoluene and propyl gallate. Let us now discuss about the third type of classification of antioxidants, which is based on the properties, that is enzymatic and non-enzymatic antioxidants. Enzymatic antioxidants are the enzymes present in our system which have the ability to neutralize the free radicals and reactive oxygen species. Some of the examples are superoxide dismutase, catalase and glutathione peroxidase. The enzyme superoxide dismutase is responsible for dismutation of superoxide anion into hydrogen peroxide. Hydrogen peroxide, which is produced by superoxide dismutase, subsequently removed by catalase. In humans, three different forms are present named as SOD1 or a copper and zinc containing superoxide dismutase, SOD2 a manganese containing superoxide dismutase and SOD3 which is an extracellular superoxide dismutase. The enzymatic antioxidant catalase converts hydrogen peroxide into water and oxygen. The enzyme is present in cytosol and mitochondria. Glutathione reductase is another type of enzymatic antioxidant. The glutathione reductase regenerates glutathione, which is a natural water-soluble antioxidant, from oxidized glutathione using NADPH as the source of reducing power. The antioxidant glutathione peroxidase has the ability to reduce hydrogen peroxide to water and also lipid or non-lipid hydroperoxides by utilizing glutathione. The other class called as non-enzymatic antioxidants include both proteins and small molecular mass molecules. Small molecules which act as antioxidants may be grouped into different categories including water-soluble, fat-soluble, natural, synthetic or endogenous and so on. Hope you can recollect that we have already seen the details of these small molecules in the other classes of antioxidants which we were discussing. If you take the non-enzymatic protein antioxidants, examples include albumin and transition metal binding proteins, in which albumin is an abundantly present plasma protein. It contributes to nearly 33% of total antioxidant activity of plasma. Through its ligand binding ability, it binds with transition metal ions like copper and iron, which acts as a source for formation of free radicals. The antioxidant property of albumin is mainly due to its ligand binding property and free radical trapping property, both of which are related to its structure. Transition metal binding proteins like iron binding ferritin, transferrin, lactoferrin, and copper binding ceruloplasmin are also examples for non-enzymatic protein antioxidants. Generally, the transition metals like iron and copper reacts with lipid hydroperoxides and converts it into reactive free radicals. However, the transition metal binding proteins chelates the metals and inhibit the formation of free radicals. Now we will see the fourth type of classification of antioxidants, which are endogenous and exogenous antioxidants, classified based on the source. As you are aware, human body is continuously exposed to free radicals which may come from outside, that is exogenous, or may be produced inside, that is endogenous. These free radicals cause tissue injury by damaging the macromolecules of the cell. Human body defends against the oxidative stress by the presence of both exogenous and endogenous antioxidants. So, endogenous antioxidants are the free radical scavengers which are produced by the cell, and exogenous antioxidants are the molecules which are obtained through diet for supplementing the activity of endogenous antioxidants. The examples for endogenous and exogenous antioxidants include antioxidant enzymes, vitamins, and so on, about which we have already discussed except lipoic acid. We will see about lipoic acid alone now. Lipoic acid is readily absorbed from the diet and acts as a good antioxidant. It functions as a coenzyme in mitochondrial enzyme complexes. The reduced form of lipoic acid, La, is called as dihydrolipoic acid, DHLA. Both lipoic acid and dihydrolipoic acid act as antioxidants and metal chelating agents. Lipoic acid and dihydrolipoic acid act as redox couple and help in the regeneration of the antioxidants like vitamin E, vitamin C, and glutathione. The last class of antioxidants are primary or chain breaking antioxidant and secondary or preventive antioxidant, which are classified based on the mechanism of action. The chain breaking antioxidants slows down or stops the oxidative process after the reaction has begun by donating or receiving an electron from the free radical species. Examples of water soluble chain breaking antioxidants include vitamin C, which scavenges hydroxyl, superoxide, and peroxyl radicals effectively. Uric acid, which complexes with iron and prevents the formation of free radicals. Glutathione, which scavenges a variety of free radicals, and bilirubin, which protects the cells from hydrogen peroxide. Examples of fat-soluble chain-breaking antioxidants include vitamin E, which effectively breaks the chain reaction of lipid peroxidation, beta-carotene, an effective singlet oxygen quencher, and ubiquinol 10, an effective scavenger of lipid peroxyl radical. On the other hand, the preventive antioxidants suppress the generation of free radicals and thus reduce the rate of chain initiation. Examples include antioxidant enzymes and transition metal binding proteins. The antioxidant enzymes decompose the peroxides so that the generation of free radicals is prevented. Examples include the antioxidant enzymes like superoxide dismutase, glutathione S transferase, glutathione peroxidase, and the endogenous antioxidant pyruvate. the antioxidant pyruvate undergoes decarboxylation and reduces hydrogen peroxide to form water and acetic acid. The transition metal binding proteins, which is also a class of preventive antioxidant, binds with metals like iron and copper and prevent the production of free radicals. Examples include ferritin and ceruloplasmin, which chelates with the metals and prevents the formation of free radicals. So far, we have discussed in detail about the different classes of antioxidants. The antioxidants which we have seen under the different classification can also be grouped into different types as enzymes, vitamins, phytochemicals and small molecules. So dear students, in this session the various classes and types of antioxidants which are available for the system to eliminate the toxic effects of oxidative stress were discussed. In the subsequent sessions, we will discuss further on the sources of the antioxidants and their physiological role in the prevention of diseases. Thank you so much for listening to this discussion on the classification and types of antioxidants.